This is Suganti, Assistant Professor, Department of Biotechnology, Vista, Chennai. In today's class, we are going to see about a Lac Operon concept, lactose operon. So before the subject Lac Operon, we must know for what we are studying the Lac Operon concept. So gene expression is the process by which the instruction in our DNA are converted into the functional product such as protein. So this gene expression is a essential biological process in all the living organism. So how the genes are expressed? For example, in prokaryotes, the multiple genes are under the control of one promoter, and we have already seen the need of the promoter and the structure of the promoter and what is the role of promoter in the transcription process. So what to transcription occur, the promoter is very important. So in prokaryote, the multiple genes are expressed under the control of one promoter, but in case of eukaryote, single gene is under the control of single promoter. So how the gene expression is controlled? You just imagine that in each cell in the human contains the genetic information, genetic material for the growth and development of a human. Some of these genes will need to be expressed all the time. So what are the genes coded in our DNA? Most of the genes are expressed in all time, but some of the genes are not expressed at all the times. So some of the genes are involved in the essential biochemical process such as expression. So those genes are expressed in all the times, but some genes are not expressed in the all the time. So those genes are must be switched on or off based on the need of the cell. So this mechanism, this is the on-off mechanism. So based on the need of the cell, the genes are expressed. So in today's class, we are going to see the on-off mechanism of lactose operon in model organism, prokaryotic model organism like E. coli. This is the genome of E. coli. Here, you can note the Lac operon. This Lac operon is expressed under the control of different genes. So what is operon? Operon is nothing but gene organization in the prokaryotes. So unlike the eukaryotes, in prokaryotes, the genes are organized into operon. So if you see the structure of the E. coli genome, you can easily find out the operon concept. That is the most of the group of genes are present under the control of single promoter like. And but if you see the eukaryotic genome, there is no operon group operon. The single genes are present under the single promoter and operator. So the operon is nothing but it's a functional unit of DNA. It containing the cluster of genes under the control of single promoter. So an operon is a group of genes that are transcribed at the same time. So if the operon containing the three or more genes, all the genes are transcribed at the same time. And also they usually control an important biochemical process. They are only found in the prokaryotes. So the operons are not present in the eukaryotes. This operon concept is invented by the Francis Jacob and Jacques Mamot in 1961. These people they have explained the gene regulation by the operon model. That means how the genes are regulated in the prokaryotic model system. Now we will see an example to see the Lac operon concept in the E. coli. For the survival of the E. coli need glucose or lactose for survival. So either E. coli can use glucose or lactose. Actually, the glucose is a monosaccharide. There is no need of a breakdown. So glucose is easily absorbable, and the organism takes the glucose directly, and no need of metabolism. But in case of lactose, it must be um, hydrolyzed. So the lactose needs to be hydrolyzed first. So the bacterium prefers. So So automatically, if both glucose and lactose is present in the environment, the E. coli first uses the easy available energy source like glucose. It won't go for the lactose. If in case there is no glucose, so the E. coli is in the condition that it's have to take the lactose for its survival. So at that case, how it's take the lactose and how it will break down, that only we are going to see in this Lac Operon concept. So this Lac operon consists of three genes, each involved in the processing of sugar lactose. So for the breakdown of the lactose, there is some gene expression needed. The genes which are involved in the breakdown of the lactose are under the control of single promoter, and these group of genes are called as a Lac operon. One of the important gene is coded for the enzyme beta galactosidase. This beta galactosidase enzyme hydrolyzes the lactose into glucose and galactose. Now the lactose are hydrolyzed into easily available energy source like glucose. Now we'll see the structure of the Lac Operon and how the lactose will be utilized when the operon on and off mechanism. This is the structural components of the Lac operon. Here you can see the group of genes Lac A, Lac Y and Lac Z. These three genes are under the control of the single promoter, and the operator also present near to the expressing gene. And in the left side you can see another gene called LAC-I. It will code for the repressor protein. And next, so what is the use of this promoter? Promoter is needed for the binding of the RNA polymerase, right? We have seen already in the transcription mechanism. Then operator. This operator only will regulate the expression of the lag. We will see in detail. So the lag genes are lag Z, lag Y, lag K. It will code for the some enzymes. These are responsible for the transport and metabolism of the lactose. Like we'll see with gene go for the which enzyme or what is the function of that enzyme. So LAC-Z gene is codes for the main enzyme beta galactosidase. It only convert the lactose into glucose and galactose. And another LAC-Y gene, it will code for the beta galactosidase, sorry, beta galactoside permease. It will involve in the transport of lactose. That means it will take the lactose outside the cell, and it will help for the cell membrane permeability. So that's why this enzyme is called as a beta galactosidase permease. Then LAC-A, this codes for the beta galactosidase transacetylase. It involved in the acetylation of the galactosides. Here you can see in detail about the structure of the Lac operon. The regulatory gene is called as Lac-I gene. It will code for the repressor protein. This I gene is expressed all the time, so it's called as a constitutive gene. So some genes are expressed sometime, but this Lac-I gene is always in the expressed state. And also this is based on the availability of the lactose in the environment. And other three structural genes LAC-Z, LAC-Y and LAC-A involved in the synthesis of the enzyme for the lactose catabolism. So catabolism is nothing but breakdown of the lactose. Here our ultimate aim is to break the lactose and utilize for the energy source of the E. coli. So in the structure of the Lac operon, there is a regulatory gene, LAC-I gene, then structural gene, three different structural gene, LAC-Z, LAC-Y and LAC-A, then promoter and operator. So how the lactose is converted into glucose and galactose with the help of the beta galactosidase. This is the biochemical reaction. Now we'll see the functioning of the Lac operon. So in the environment, there is no glucose, only lactose is present, or lactose is absent. At the time, what happened to the operon? Whether the operon will be in on condition or off condition. So in case of lactose is absent, the operon will be off. Here you can see in the diagram, you can see In the absence of the lactose inducer, the regulatory gene produces the repressor protein, that is Lac gene. Lac gene produces the repressor protein and it will bind to the operator site. So, and it will prevent the transcription as a result. So, here you can see uh, there is no lactose in the environment. Okay, here you see there is no lactose in the environment. That is the case is called as in the absence of lactose. What happened to the operon? Okay, so this repressor lac gene is always expressed. It will produce the repressor protein. So in case there is no lactose, it will directly go and bind to the operator. Here you can see the same repressor is present here. So automatically it will block the way of transcription. That is the promoter, the RNA polymerase, it won't go further to transcribe the LAC Z, LAC Y and LAC A. So automatically there is no expression of the structural gene of the LAC Operon. So there is no expression of the enzymes required for the lactose metabolism. So there is no need to produce the enzyme, and also there is no lactose in the environment. So based on the environment condition, the operon will work. So in case of the lactose is present in the environment, now what the E. coli have to do? It must break the lactose and it must convert the lactose into the uh, easily available energy source like glucose and galactose. Now the operon must be turned on or automatically it will turn on. Okay. So when the lactose is present in the environment, it will introduce in the medium, it will bind to the repressor. So always the repressor will be synthesized from the lac gene. And what happen in case the lactose is present in the environment, it will go and bind to the repressor protein and it will stop the further binding of the repressor to the operator. So now the operator is free and it will induce the RNA polymerase to bind to the initiation site and the promoter, and automatically it will continue the transcription process. Here in this picture, you can see in the presence of lactose in the environment, here the lactose molecules are present in the environment. So the now the cell is in the E. coli cell is in the condition of it have to utilize the lactose and it have to break down the lactose. So automatically based on the situation, the genes are expressed. Whether the lactose is present means it need to be need to be catabolized, right? So the automatically the genes are expressed. In case there is no lactose, there is no need to break down the lactose. So the genes are suppressed. So here the lac gene is produce the repressor protein. But what happened means in case the lactose is present in the environment, the lactose it will go and bind to the repressor protein and it will form a lactose repressor complex. So what is the function of this lactose repressor complex? Normal repressor only it will go and binds to the operator. In case it, any structural modification the repressor means it won't do its action, right? So there is no binding of the repressor to the operator. So here the normal transcription or gene expression will take place normally, like binding of the RNA polymerase to the promoter, and it will go further like to transcribe the LAG-Z gene to produce the beta galactosase and transcribing of the LAC-Y beta galactosase. So all the enzymes respond needed for the lactose catabolism will be synthesized by the operon, and it will easily break down the lactose into easy available glucose and galactose. So this is the mechanism of lac operon. So in this, we must differentiate when the lactose is present in the environment, the operon is off. When the lactose is present in the environment, the operon is on. If there is no lactose in the environment, the operon is off. That is the concept of the lac operon. Thank you.